My name is Britta Lindquist. I'm a postdoctoral fellow in Jean Paz's lab. I came to Gladstone because I wanted to work with Jean Paz. I knew of her reputation as an electrophysiologist and as one of the pioneers of optogenetics and genetic ablation that I knew were going to be really critical for understanding the initiation, maintenance, and termination of seizures. I love working at Gladstone. It's full of people who are at the top of their field, and yet it's small enough that people are approachable, and you can just be walking by a bench and strike up a conversation with somebody. Like, for example, not long ago, Neil Bennett and I, Neil's in Ken Nakamura's lab, started talking about his work on ATP quantification in living cells, and it led to a whole series of really exciting pilot experiments and collaboration. I decided to go to medical school after spending a couple of years after college working in a special needs classroom in the Munich International School in Starnberg, Germany. I was working with kids with all sorts of learning differences, some with language processing disorders, others with fetal alcohol spectrum disorder, a lot of dyslexia. And I realized how rewarding it was to work with people on the concrete day-to-day challenges that they encounter in, just in daily life. And that was my first clue that I might enjoy clinical neurology. So I first went to medical school intending to be a pediatric neurologist, which is not how it ended up turning out, but I'm still very grateful to my time working with students over at MIS. My current research project is all about seizure termination. Something like 1 in 10 people in their lifetime is going to have a seizure, and a significant number of them go on to develop recurrent spontaneous seizures and epilepsy. Most of the time, a seizure happens, it lasts 30 to 60 seconds, and it ends on its own. And everybody just breathes a sigh of relief that it's over. And so much of our epilepsy research is focused on the initiation of seizures. Where do they come from? How do they start? With an eye towards surgical treatment or calming down the circuits so that seizures won't start. A significant minority of seizures actually don't stop on their own. They go on to develop into status epilepticus, and that's a neurological emergency. Untreated, it can cause brain damage. And so I'm really curious about why it is that most seizures do stop on their own. You can imagine that there must be multiple redundant mechanisms, which fall into a few different categories. Maybe most exciting for me, based on my background, it turns out that people are starting to observe that a seizure can lead to feed-forward excitatory activity sufficient to trigger a spreading depolarization, which acts like a circuit breaker on the seizing network and inactivates voltage-gated sodium channels and really turns off synaptic transmission through a variety of mediators. I first became fascinated by epilepsy when I was a neurology clerkship student, so in my 4th year of medical school, when I was starting to make rounds on the wards and meet patients. I saw a patient who was post-ictal, meaning he had had a seizure and it stopped on its own. But his thinking and his communication were not back to normal, so he was really aphasic. I remember my preceptor pointed to her coat and asked, "Is this white or green?" And he picked green. And I remember she held up her knuckles and she said, "What are these called?" And he couldn't come up with it. And she said, "Are they nickels, knuckles, or buckles?" And he chose buckles. And it just blew my mind that such a large territory of his left temporal lobe and frontal lobe must have been involved in some process that just shut off all sorts of activity. And the idea that you had such a profound suppression of network circuitry, and yet, given time, his symptoms would clear and he would be able to communicate normally, and that there was no permanent tissue damage, was just really fascinating to me. And I wanted to understand more about how that network was modulated by the seizure or the processes that turned off the seizure. I love to play outside, so running, biking, hiking, swimming, climbing, anything that I can do to get fresh air and sunshine and enjoy the Bay Area. If I could meet any scientist from any point in time, I'd like to meet Marie Curie. I read her biography written by her daughter Eve, and she just seems like such a humble and yet dynamic scientist. She was totally lucid and like unforgiving in her experimental design. She valued data and experimentation over theory, and her notoriety was a tool that she used to shine light on the plight of her fellow human and to effect policy change that was for the greater social good.